Hi friends! Today we are going to speak about cholinergic drugs, but first I want you to know what the parasympathetic nervous system is and how it relates to these drugs. The nervous system is the system that predominates in a quit, rest and digest condition, and this system has some effects on almost all the body parts, such as stimulating bile release, decreasing heart rate, bronchoconstriction, stimulating salivation, meiosis, bladder contraction, stimulating peristalsis, and secretions. The parasympathetic nervous system uses acetylcholine as a neurotransmitter for both pre- and post-ganglionic neurons. And as we know, acetylcholine is the neurotransmitter at neuromuscular junctions, at synapses in the ganglia of the visceral motor system, and at a variety of sites within the central nervous system. Acetylcholine originates from two major places in the brain: 1. The basal forebrain, and 2. The mesopontine tegmentum area. And it originates in the basal forebrain from the basal nucleus of Meynert, the medial septal nucleus, and the nucleus of the diagonal band. Dear listener, there is something very important that you should know, which is that acetylcholine is non-selective, which means it will switch all cholinergic receptors in the body. And also, it is easily hydrolyzed in the blood with esterase enzymes, which means it has a short half-life or a short duration of action. Plus, it cannot be taken orally because it is easily hydrolyzed by acid catalysis in the stomach. Now let's delve a little deeper together so we can get to know this neurotransmitter in some detail about the structure-activity relationship. At first, we will divide it into three important parts. The first part is the quaternary ammonium ion, and you should know that 1. The positively charged ammonium ion is essential for activity. 2. And replace N with arsenic, phosphorus, or sulfur, decrease the activity. 3. In addition, there must be 2 methyl groups at the nitrogen atom, at least. 4. Plus replacement of 3 methyl groups with larger groups will lead to a loss of its agonist activity because it can't fit its active site in muscarinic receptors. The second part of the acetylcholine structure is the methylene bridge. 1. According to Ing's rule of 5, there should be no more than 5 atoms between nitrogen and terminal hydrogen to form a maximal effect. 2. And extending the ethylene linker length decreases the activity. 3. Addition to substitution on the ethylene bridge. As for the alpha carbon, which will give acetyl A-methacrylene, it is less potent. Or beta carbon with methyl, which gives methacrylene selective action on muscarinic receptors. Here we come to the last part of the acetylcholine structure, and it is the acyloxy group. 1. According to the rule of 5, replacing acetyl with higher molecule homologies, such as propionyl or butyryl groups, will give a less potent analog. 2. Plus the aromatic or higher molecular weight esters will antagonize the activity. 3. And the NH2 group instead of the acetyloxy group is more stable to hydrolysis and increases the duration of action. As we know, acetylcholine has no selectivity of action. That's why it will switch on all cholinergic receptors in the body. But now we will go through the binding site of muscarinic receptors. We should know that muscarinic receptors have 3 hydrophobic pockets, which will be connected to acetylcholine. The quaternary ammonium ion is connected with ionic bond, and the acyloxy group binds with 2 hydrogen bonds. Okay, now I think you wonder how this acetylcholine is formed inside your body. So I'll answer your question by telling its biosynthesis in our body. In our bodies, we have many amino acids. One of them is serine, and by serine decarboxylase will give 2-aminoethanol, and then with N-methyltransferase, or S-adenosylmethionine, which is a very important methyl donor, so we'll add 4 methyl groups on NH2. Now we have choline, and by glycolysis in body, give acetyl-CoA enzyme, which will convert choline to acetylcholine. After all this, I'm sure that you already know the importance of sufficient amount of acetylcholine in our bodies. So, the deficiency in acetylcholine may develop some common diseases such as myasthenia gravis, Alzheimer's disease, Parkinson's disease. Now it is our role as pharmacists to find drugs for these diseases, and these drugs are cholinergic drugs. Cholinergic drugs or parasympathomimetics can be classified according to their mechanism of action into 2 categories: direct and indirect. So we will talk about those categories in some detail. 1. Direct-acting or cholinergic agonists work by binding directly to activate muscarinic receptors, and when a neuron in the parasympathetic nervous system is stimulated, it releases acetylcholine, which crosses the synapse to interact with the receptors in the adjacent neuron. So simply, cholinergic agonists stimulate cholinergic receptors, mimicking the action of acetylcholine. Direct-acting cholinergic drugs or cholinergic agonists can be subclassified into two types: 1. Choline esters and 2. Plant alkaloids. 1. Choline esters are structurally similar to acetylcholine, like bethanechol, methacholine, and carbachol. 2. Plant alkaloids, like muscarine and pilocarpine. Indirect-acting cholinergic drugs can also be subclassified into 1. Reversible inhibitors of the acetylcholinesterase, and 2. Irreversible inhibitors of the acetylcholinesterase. Bethanechol. It is choline ester, direct-acting cholinergic drug, so it mimics the action of acetylcholine by being selective agonist on muscarinic receptor. It is primarily used to stimulate smooth muscle contractions, especially in the gastrointestinal and urinary tracts. So it has selective action on urinary bladder and gastrointestinal, plus it promotes bladder emptying and gastrointestinal peristalsis. Bethanechol is approved by the FDA to treat postoperative urinary retention, postpartum urinary retention, and overflow incontinence caused by neurogenic atony of the bladder. And now, we will know the structure-activity relationship of it. But first, it is important to note that the SAR of bethanechol is so close to the acetylcholine SAR. I think you already have noticed the similarity between them. And the second thing that you should know is that it is important to focus on its interactions with muscarinic receptors and smooth muscle tissues. Modifications to its structure should be carefully designed to maintain the desired agonist activity while minimizing potential adverse effects on other receptor systems. So we will show the parts that are different from acetylcholine and have an important role as well. The first part is the methyl group at beta position. That makes it selective for the muscarinic receptor, and it forms a steric shield to protect the drug from estranged enzymes, so slow down this chemical and enzymatic hydrolysis and increase the duration of action. The next part is the NH2 group instead of the acetyloxy group of acetylcholine, and it makes the drug more stable for hydrolysis, so longer duration of action as well. That's due to the lone pair of electrons on it, which makes resonance with the carbonyl groups. So in this video, we have learned about the parasympathetic nervous system and how it affects the body, the neurotransmitter acetylcholine and its structure-activity relationship and its binding site on the muscarinic receptors, and its biosynthesis inside the body. The cholinergic drugs, and we talked in detail about the directly acting mechanism, and as an example, we talked in detail about bethacholine. Thanks for your time. I hope you found that helpful and insightful.